laser on human cells. Is that correct?
Yes, exactly.
Okay, and with the lifespan research on the worms,
Sorry.
No, hey, that's no problem. We, hey, we're grateful.
Okay, don't play. It's okay. Okay.
Hey, Nason, that is no problem. We the show here tonight. We are always grateful for any guest that we can have on the show, and whatever time you can make for the show. So, hey, people, kids, families, dogs, we've had them all on the show before, so don't you worry one bit. I was asking about the lifespan test that you were going to do on the worms. Could you give us a little description of perhaps maybe one or two different laser treatments you're going to try out?
Yeah, first of all, we're going to start with what we think are close to the optimal parameters for destroying pigment granules in general. Now, the worms are going to be a little different because they have some smaller pigment granules because they have smaller cells. Everything's kind of scaled down.
Okay.
But we're basically going to start with our best guess of what the closest to optimal treatments as far as the length of the pulse and things are going to be, and that'll be like a first run. And of course, along with that first group, we'll vary a couple of those parameters. Maybe try a little bit longer pulses and things like that to try to see if there's any difference in how we're affecting lifespan. But this of course, is after we do a preliminary experiment in which we verify at the given age we start treatments what power levels are going to kill the worms right out. So you find that basically, your LD50, that's the level at which half of your animals are killed by a certain power level.
Okay.
And that gives you a starting point. You say, okay, we'll sneak under that a little bit and try a range of power levels and see how that's affecting lifespan to make sure you're not just causing damage that's going to outweigh any benefit you do.
Right.
So that's kind of our first two experiments. You do your initial power level adjustment, and then you take your best guess at parameters with a couple variations and see what kind of effects on lifespan that are there, and make you organize your next experiment accordingly.
Okay. Basically. So and you're going to use the roundworms now. From what I've read, what is the typical lifespan of a roundworm? 15, 20 days? Something
C. elegans, they'll live 15 days on average if you grow them at 25 degrees Celsius, about 77 degrees Fahrenheit. It's very temperature dependent. If you go down to where I'll be working at, just because of the equipment we have, I'll be working at 20 degrees Celsius, which is about 68 degrees Fahrenheit. And they should live a little longer. They should have an average lifespan of about 19 or 20 days. So that'll also help us see some finer details if we're only having a minimal effect with our initial treatments.
Okay.
We'll be able to see some subtle differences there to give an idea of how we should modify things.
So the idea is to give the laser treatment to young worms who have just a few lipofuscin granules, and hopefully that will get rid of that lipofuscin, and then you'll continually do that to find out how long the lifespan can be extended through this procedure, if at all. Right.
Right, and that's my best guess for the mildest type of treatment that we can do. Obviously, what we want to be able to do is say, look, we can take an organism that's 90% gone and bring it back, and have this great rejuvenation property. But if we can do that, it's obviously going to be very more difficult and require a high degree of fine-tuning parameters. So we want to do the easy task first. And that's going to be treating young organisms that can handle maybe losing a cell or two and still have a fair lifespan. Especially in these worms, you kill too many cells and they just can't handle it. But it's a good proof of concept, because if we can get them to live even a little bit longer with the treatments, then that bodes really well for more complex organisms that can afford to lose a lot more cells.
Sure. But then you do you hope to at least try some of the laser treatment on older worms as well, right?
Yeah, definitely. I mean, especially if we're getting positive results on the younger ones, first thing we're going to do is
move right up.
We'll do like 5 or 6 different groups starting at various ages to see what kind of effect there is depending on the age of the worm.
And I'm going to mention here that something I didn't mention earlier is that Nason Schooler will be working with the SENS Foundation in Arizona at the Institute for Biomedical Gerontology in conducting these lifespan experiments. And for those of you not familiar with the SENS Foundation, they are working on all types of engineering, regenerative engineering experiments and processes to hopefully rejuvenate eventually the human, not just worms. and that brings me to the next question here that I have, and that is, what type of experiments will you be doing on human cells? I assume, I'm just thinking here that you're going to have some live human cells that have some lipofuscin pigment, and you're going to blast them with the laser or tune the laser to see if you can get rid of the lipofuscin in these human cells in culture.
Yep, exactly, and that has several benefits because the cells are larger, the granules of lipofuscin can get a lot larger, so we can see under the scope directly what kind of effect we're having on the lipofuscin. We'll be able to see the viability of the cells, how many cells are we killing with the treatments, can we do treatments that don't kill cells at all and still eventually wear down the lipofuscin. We'll be able to do a lot more fine-tuning type experiments like that with the human cells.
Right, and that's what I was thinking. Perhaps we go in hopeful and optimistic that we can tune the laser to selectively destroy lipofuscin. But even if it proves tough in human cells, I expect that we'll learn something very interesting about how the laser breaks it down and how the human cell, digests it, if at all, afterward.
You bet we will. And I'd like to note that there is some very encouraging preliminary literature on this subject. For instance, in ophthalmology, they have used pulse lasers to selectively destroy lipofuscin. It clears it out of the tissue is rejuvenated, and it's basically back to where it was before the accumulation of lipofuscin was a problem. Now, of course, these cell types, they're RPE cells, they're about the best cells you could hope for. I mean, they're post-mitotic, so they're kind of accumulating lipofuscin and they're not replacing each other until they're damaged. And since it's in the eye, since it's in an important tissue like this, they are capable of repopulating.
So,
even though you kill the older cells.
I'm not familiar with the research, but this is for macular degeneration, correct?
Yes.
And these cells, now with the lasers that they use to clear up macular degeneration, those cells, as far as they know, are dying when they do the laser treatment?
Yeah, but they're doing overkill. I mean, the whole use of lasers. Yeah, the whole use of lasers in ophthalmology started out backwards. It started out saying, wow, we can use this laser and basically melt this tissue and fix some problems, in areas of like retinal detachment and things. And they could actually have some benefit from completely coagulating portions of tissue and stuff. And so basically it was just built upon that. They're like, oh, what different parameters can we try?
They knew that the cells would rejuvenate, so they didn't have any fear of overdoing it and just destroying
no, actually, when you have coagulation and scarring like that, even in a tissue like the RPE, you get to a point where there's too much scar tissue.
Oh.
It can't reorganize itself correctly. So ophthalmology, they kind of started out with outright damage and, gee, what can we do with the damage? And it's only been in maybe the last 5 years or so where they've really seemed to have gotten into fine-tuning pulse parameters or quickly scanning laser beams to achieve the same effect to get more selectivity. Because with the pulses, that's what you get. You get more selective heating of the pigment granules themselves.
So I'm thinking that your research will also have applications then for macular degeneration. I would think.
Of course, and imagine if we got to the point where we're applying this a whole body exposure of different levels of this laser radiation. Sure, you're gonna hit the pigments in the eye. You're gonna hit pigments involved in Parkinson's and Alzheimer's disease in the brain. I mean, you're probably going to be hitting some. Well, yeah, it depends on how you set your parameters.
Sure.
But
right.
yeah, as far as